really clear. Oh, 10. Okay. Thank you. Good morning, everyone. Here, I would like to share my research with title of research about invasive species. Especially in here, I make a title first record of sea bass peacock. There are genus Cycla ocellaris from the Gedung River in Yogyakarta, Indonesia, revealed by DNA barcoding. I'm Sabta Andriono from Department of Marine and from Faculty and Marine Fisheries and Marine Faculty Universitas Erlangga, and some collaborator from the Balai Karantina Yogyakarta, and also my colleague in Bangladesh. And this is my outline of a presentation from introduction until conclusion. Is very brief, so I hope any discussion, if not clear or not completely explain of all my results. As the introduction, we can see that in Indonesia, we know many fish, especially for ornamental fish, now introduced from the abroad, especially from America or Africa. But now from introduction fish will be some problem in Indonesia that become an invasive alien species, or we can abbreviation with the IAS. And approximately 39% of the fish already distinct and over the 400 years. It's very dangerous for biodiversity in Indonesia. So currently, this research focuses on invasive species in Indonesia, which called Cycla or Sibascoc, which distribute almost in Indonesia, especially for ornamental fish. And this work initially by Yogyakarta Fish Quarantine of Fish Quality Control, or Balai Karantina Jogja, we already have data on the survey on August and October 2020, which able to get this fish in the river, Gedung River, Jogja. And this research, we use the molecular approach for the identification. First, we collect sample in the downstream and upstream, because the location is very suitable for the habitat of the Cycla. And here the coordinate location. And beside the molecular identification, we also conducted based on morphological, even just in the pattern of the color. And then we extract DNA and then PCR condition according to the kit of DNA extraction kit. We use AcuPrep genomic DNA kit and use FISH PCL and BCH primer, because this is a universal primer for fish. And we use a traditional PCR using 20 microliter with component including enzyme and etc. We can discuss more if any interest about this process. And then after get the results, we also use some bioinformatic software. And here we use Mega 10 software to make a phylogenetic tree and another data analysis. And for raw data of sequence, we use chromas to align and also to trimming the bed segment of the segment of DNA. And then some open source of bioinformatic program like a reverse complement and also Clustal Omega to align the sequence to make a valid. And here the result of our finding about Cycla species. At the location, we only get 7 sample with the different size, and we cut the caudal fin to extract of DNA in the small part to identification of base molecular. But we also collect some information morphological, including the pattern, even not completely clear. And also we collect the information about habitat, because in here very good and very suitable for the freshwater fish habitat. We can see the upstream very good with the high dissolved oxygen, and also pH of the water is also still normal. In here is very good for local or native species of freshwater, but at the time we cut the invasive species like Cycla species here. And after molecular identification approach, we successfully amplify the sequence from the sample. We got more than 500, and after alignment, 523 base pair, able to adjust that species. Almost all species is a Cycla ocellaris from 99.68% to 100%, with the gene bank accession number of the reference is KU878410. And we also conducted phylogenetic analysis that all sample similar with the reference Cycla ocellaris from the Malaysia, and including a very same as genetic sequence with the Cycla temensis. In here, we can see that the phylogenetic tree show the Cycla temensis and ocellaris in the same line. So it's mean maybe I have a same genetic, but from the morphological pattern, it's show that based on reference Supra Pravita as also 2020, that ocellaris have very unique slots or bar in the lateral side. So we can see the sample also close to the ocellaris, then temensis is distinguished and then make what we call the valid for the identification based on molecular that we adjust the species is Cycla ocellaris. And we can see the distribution of many Cycla around the world. From the South America, they distribute almost around the world, and in Indonesia we can see that dominated by Cycla ocellaris here, including in Malaysia, and also some region close to the Indonesia is number 17 is Guam, in the close to the Papua. But this species is most dominated. But we from several paper previous research about the Cycla explained that the genus Cycla is probably they make a genetic mix between species, because some genus maybe mix breeding, so make a new pattern and then maybe new variant of species. It's possible to study more deep because the it's not only just identification species, but we can use another region gene to identification more detail about the variant. And here, unfortunately, I'm not conducted the DNA from nuclear, but only from mitochondrial gene. So just for identification. And the last part of this presentation is we conclude that our sample is species of Cycla, and then this species is from Africa. Then distribution of this species until Jogja, and maybe in this in another region of Java, based on because this species is the ornamental fish, and some area used for fishing attraction. So like hobby fishing in the some area, and then sometime release naturally in the river or the dam. So we suggested that the government and also scientists are more doing collaboration to monitor of the non-native species in Indonesia. So hopefully our biodiversity will be conserved, and then we still keep the genetic of the local species, especially Indonesian fish, not contaminated by the invasive species. And that's all my presentation, and I would like to deliver my knowledge our collaborator from station Fish Quarantine, Quality Control and Fisheries Yogyakarta, or Balai Karantina Yogyakarta, and also my colleague in Department of Fisheries, Ministry of Fisheries of Livestock, Bangladesh, with a very fruitful discussion during preparation of the manuscript. Thank you so much. I hope any discussion from this room 3. Thank you. Back to moderator.
Thank you, Mr. Sato, for interesting presentation. Next, we will continue to the second presenter is Mrs. Asna Sofia Milan. Hello, Mrs. Asna.
Yes, hello. May I present my own PPT?
Yeah, whether the slide presentation come from us or directly from you?
Yes, from me.
Oh, yeah. Okay, thanks. Mrs. Asna.
Okay. Is my screen, you can see my screen?
Yeah. Yeah. Okay, start your presentation, please.
Okay, the Honorable ISFFS Committee, participants, and all of my friends, good morning. I'm Asna Sofia Mila, the presenter from Echinoderm Scientific Class, Kelompok Studi Kelautan Universitas Gadjah Mada, and I will present our research entitled Diversity of Echinoderms from Intertidal Zone in Kukup Beach, Gunung Kidul, Special Region of Yogyakarta. This is the table of contents that I will talk about. To begin with, I want to show the substrate that we took our sample. From the right picture, we can see the substrate in day, but we took our sample at 3 a.m. So in the left side, this is the picture of the beach. For the background, the proliferation of flora and fauna as components of biodiversity also happens in marine ecosystem. Indonesia is comprised of 78% seawater between the Pacific Ocean and the Indian Ocean, with significant population algae, corals, mollusks, sponge, echinoderms, etc. Kukup Beach is scarce beach located in Gunung Kidul Regency, Yogyakarta, and this beach is one of the habitats that supports the diversity of echinoderm species in Indonesia. Echinoderms is one of the benthic animals and has a habit of hiding on rocks in intertidal zone, and echinoderms play an important role in ecology. The purpose of this study is to record the diversity of echinoderms in intertidal zone of Kukup Beach, Gunung Kidul, Yogyakarta. Moving to the methods, this study was carried out at intertidal zone of Kukup Beach, Gunung Kidul, Yogyakarta on March 10, 2023 at 3 to 5 a.m. Western Indonesian time during maximum low tide. And this is the equipment and materials. From the figure 1, we can see the satellite image of research site in Kukup Beach, Gunung Kidul. So we divide into 5 transect A, B, C, D, E, and place the plot vertically toward the sea, so we can expect to get more diverse species of echinoderms. This is the method. So the sampling conducted at intertidal zone. Data collection was conducted using a systematic sampling method equipped with 1 meters times 1 meter squared plots and place 5 transect A, B, C, D, E. We measured the physicochemical parameters and we identified the species that we have collected. Moving to the result and discussion. So I will talk about the characteristics and human activities of Kukup Beach. Kukup Beach has wide sands and substrate that compose of dead corals. The area becomes deeper with dead corals as you move towards the south, and it falls under the intertidal zone, revealing marine organisms in the hollows of dead corals during low tide. And this beach experiences high levels of human activity, including tourism and the utilization of marine resources during low tide. This is the physicochemical parameters. So for the air temperature is 22 degrees Celsius. For the water temperature is 26.80 degrees Celsius, while the optimum is 26 to 30. And the pH level is 7.97, while the optimum is 7.5 to 8.6. And the salinity is 34 ppm, while the optimum is 30 to 36. So as we can see, we can conclude that when we took our sample is in optimum range of physicochemicals when the echinoderms can live. For the air temperature, we cannot find the literature because air temperature not really affected the diversity of echinoderms. And this is the echinoderms that we found in Kuku Beach, consists of two classes, Echinodea, Oviridea, free family, Echinometridae, Stomopnestidae, Opiocomidae, and 6 species. The first one is Echinometra matei, that have round, slightly oval, and black body shape. The body temperature is 3.5 centimeters and body height of 5 centimeters. It has a pale ring and the base of its spines. This species distributed in tropical area found on reefs in the Indo-Pacific region. The second species is Heterocentrotus trigonum, has a diameter of 9.5 centimeter with blunt and thick thorns. And the species known as the pencil sea urchin due its spine and has radius-prone species in areas with the dead coral reefs and rock that have relatively large holes. The third species is Stomopneustes variolaris. The species has black body, oval-shaped shell, body length 12.5 centimeters, and upright spines. The habitat of this species in rock pools, crevice, holes, and river-shaded area with constant water circulation. The species distributed in the Arctic to Antarctic region and in all tropical oceanic waters in the Indo-Pacific region. The fourth species is Ophiocoma scolopendrina. The species has 10.5 centimeter length and a diameter of over 20 millimeters, and there are typically no more than 4 arm spans basally. The discs are coarse with granulation, and uppermost arm spines thickened and cigar shaped, rarely tapering. The species has lighter body color underneath and darker on the upper part of its body. The fifth species is Brevitur macrohi. This species we can find dark brown star shape and the arbor side with some white and light brown pattern on each cap set type. And on the arbor arms, there are light brown and dark brown strip line patterns. And according to Bayesian, the species has spine that can be found on its segments. This characteristic becomes one of the diagnostic characteristic. And we can find the species in Indian and Pacific Ocean and lives on coral reef with depth between 0 to 30 meters. This is the sixth species, Ophiocoma erinaceus. The species has a distinctive star-shaped body with long and slender arms. It can grow up to maximum size about 20 centimeters in diameter and including the arms. The coloration of this species can vary, but it typically has a mottled pattern with shaded are of brown, gray, or reddish brown. And this species distributed in the Indo-Pacific, Maldives, Eastern India, Northern Australia, and also the species can be found in south of Balticus, the cluster of Pulapari, Thousand Island, Jakarta. Moving to the conclusion, so from this research, we can conclude that there are 6 species of echinoderms found in Kuku Beach. For the Ophiuroidea class, there is Ophiocoma erinaceus, Ophiocoma scolopendrina, and Brevitur macrohi. And for the Echinodea class, there is Echinometra matei, Heterocentrotus trigonirus, and Stomopneustes variolaris. The calculated parameters value were air temperature 22 degrees Celsius with a temperature 26.80 degrees Celsius, and the water pH is 7.97, and salinity is 34 ppm. So this is reference that we study. That's all from me. Thank you for the attention. I will hand back the time to the moderator.
Thank you, Ms. Asna, for the interesting presentation. Okay, for the next presenter is Laila Widi Utami. Operator, please set up the presentation file.
Oh, dia mau.
Yeah. Okay, Miss Laila Widiutami, time is yours.
Here I am here to represent Laila Widiutami as the presenter. And good morning for all of the audiences. I'm Alvista Diska here to present about my team's research entitled Macroalgal Abundance in Intertidal Zone of Cocoa Beach, Gunung Kidul, Yogyakarta. Next. The first one is for the introduction, and we know that macroalgae divided based on their photosynthesis pigment into rhodophyta, chlorophyta, and phaeophyta. And there are several environmental factors that affect the macroalgae abundance, which is temperature, pH, light, nutrients, salinity, dissolved oxygen, and anthropogenic activities. And this research was conducted at Cocoa Beach, Gunung Kidul, Yogyakarta, Indonesia, with dead coral fragments, and white sand substrate. And abundance of macroalgae can be influenced by its physicochemical factors, and the changes in the physicochemical factors can change the condition of macroalgae abundance. Next. And based on the introduction, this research was conducted to study the abundance of macroalgae in Cocoa Beach and measure the environmental parameters that affect macroalgae abundance. Next. And for the methods that we use in this research is first for the sampling location and time. And the study was carried out on March 11 at the intertidal zone of Cocoa Beach, Yogyakarta at 3 until 5 a.m. Western Indonesian time during low tide. And as we can see in the figure number 1 about Cocoa Beach appearance from Google Earth. Next. And for the materials and equipments that we use in this research is 5 quadrat plots with the size of 1 by 1 meter quadrat, raw meter, pH meter, thermometer, refractometer, and camera. And for the materials that we use are macroalgae samples from Cocoa Beach and identification books named FAO, volume number 1, Species Identification Guide for Fishery Purposes. Next. And for the sampling method, and first is the sampling area was determined by measuring the overall intertidal zone of Cocoa Beach during low tide, and the sampling area should cover up at least 1% of overall intertidal location size. And the sampling zone was divided into 5 vertical transects with 9 sample areas for each transect that we can see in figure number 2. And overall, there are 45 sample zones that we use, and data collection was done by recording the subplot for each sampling point using morphological observation. Next. And next is for the identification. The documented samples are then by book entitled FAO volume number 1, Special Identification Guide for Fishery Purposes, and for the additional source such as previous research, documentation, and expert opinion were also used to confirm the identified species. And for the data analysis, we use quantitative data analysis was conducted to determine the coverage of each macroalgae species that was found in the sampling area. Besides, we use species cover-up frequency, relative frequency, density, relative density, and important value were counted. And beside that, the environmental parameters which have been measured are also being counted using software called Microsoft Excel program. And then the quantitative result was analyzed. Next. And next is for after the research and sampling that we did, we got two results. The first one is the species importance value index of macroalgae in Cocoa Beach that we can see in table number 1. And based on the sampling and data analysis, macroalgae abundance in intertidal zone of Cocoa Beach, Gunung Kidul, Yogyakarta, we got 23 species of macroalgae, which are divided into 18 family, which is Rhodomelaceae, Dasycladaceae, Cladophораceae, Buddleaceae, Codiaceae, Ulfaceae, Solieraceae, Gelidolaceae, Gelidiaceae, Gracilariaceae, Siphonocladaceae, Acrotilaceae, Rhodomelaceae, Dictyotaceae, and Palamariaceae. And based on the result in table number 1, it can be seen that the macroalgae species in Cocoa Beach which have the highest relative density and relative frequency values, respectively, was Cladophora columbiana species with the order Cladophorales and family Cladophora CA, with the relative density is 21.10% and relative frequency is 9.09%. And meanwhile, the species with the lowest relative density and relative frequency values was Bonetella nitida of the order Decicladales and family Decicladaceae at 0.09% and 0.57%. Next. And for the second result, we can see in table number 2 about physicochemical factors. And based on the result in table 2, it can be seen about air temperature, water pH, and water salinity averages, and that the air temperature is 2020 Celsius degree, with water temperature is 26.80 Celsius degree, with standard deviation is 0.837, and water pH is 57.97 with a standard deviation 0.049, and water salinity is 34 ppm with standard deviation 8.124. Next. And for the discussion is the distribution of macroalgae in Cocoa Beach is dominated by Cladophora columbiana, as you can see in figure number 3. And Cladophora columbiana can grow well in Cocoa Beach because the substrate types are sand and coral, which are suitable habitats for macroalgae growth as well. And Cladophora columbiana distributed well on coral substrate in shallow waters exposed to high sun exposure. And the species with the lowest abundance and distribution is Bonetella nitida, because usually Bonetella nitida can grow well on sandy or muddy substrate around the lower intertidal zone in moats or shallow lagoons with high sediment and nutrient. And for the abundance of macroalgae is influenced by aquatic environmental factors such as air temperature, water pH, and salinity levels. And besides that, their substrate can significantly affect the abundance of macroalgae too. Next. And for the conclusion that we can conclude for this research is there are 23 species of marine macroalgae found in intertidal zone of Cocoa Beach, Gunung Kidul, Yogyakarta, and Cladophora columbiana is a macroalgae species that dominated the sampling location, and the species with the lowest dominating index is Bonetella nitida. For the environmental parameter analysis shows that the water temperature, salinity level, and pH level can significantly affect the growth of marine macroalgae species. Next. And this is for the references that we use in this presentation. Next. Thank you for your attention. I'll bring it back to the moderator.
Thank you for Ms. Laila. Okay, for the next presenter, it's also you. You will presentation, yeah. So you can share your PowerPoint for the second presentation. Okay, Ms. Laila, time is yours.
Okay, thanks for the opportunity. Good afternoon, dear ISFS committee, and to all the participants in this room. I am Aisa Saidina, and I will present a research that we titled on the biodiversity of macroalgae at the Cocoa Beach, Yogyakarta, Indonesia. Next. Okay, first, for introduction, macroalgae are a group of multicellular algaeous bodies or cells which do not have true roots, stems, and leaves. Macroalgae are part of a heterogeneous group of photosynthetic organisms. One way to classify macroalgae is based on their pigmentation, which is divided into brown algae or pyophyta, green algae or chlorophyta, and red algae or rhodophyta. And the characteristic of brown algae are xanthophylls and fucoxanthin as the dominant pigment. Distinctive feature of chlorophyta are the presence of chlorophyll A and B acids. And as for the pyophyta, it has characteristics, namely beta-carotene, chlorophyll A and C, as well as xanthophyll pigment. And for the rhodophyta, below protein pigments predominant, along with the other pigments such as chlorophyll, A, B-carotene, and xanthophyll. And in the environment, algae have an important role as the main source of food, raw materials for the manufacturers of polysaccharides, fertilizers, antibiotics, biological research, and waste management. Next. So this research was conducted in the intertidal zone of Kuku Beach, Gunung Kidul, Daerah Istimewa Yogyakarta. And Kuku Beach has rocky characteristics and big waves because it is directly attached to the Indian Ocean. And for the activities are usually carried out by tourists when the water recedes on the beach. And the intertidal zones of Kuku Beach substrate is dominated by rocky coral and covered by macroalgae. Next slide. Okay, although the little research has been done on the diversity of algae on Kuku Beach, this research was aimed to studying the diversity of macroalgae in the intertidal zone at Kuku Beach and determining environmental factors that affect distribution and diversity. Next. So this research was done in March 11, 2023 at Kuku Beach, that located in Kemadang Tanjung Sari, Gunung Kidul, Daerah Istimewa Yogyakarta, and this research was conducted at 3 to 5 p.m. in the intertidal zone during low tide. And next slide. And this is a picture of intertidal zone of Kuku Beach through Google Earth application. Okay, next. This is for the method, the materials and tools that we use in this study are macroalgae sample from Kuku Beach and identification books such as PAO Fall 1 Species Identification Guide for fishery purposes. And for the equipment that we use in this study are 5 product plots with the size of 1 cube meter, raw meters, pH meters, thermometers, refractometers, and camera. Next. And this is the step of the research. There is a 3-step, including sampling of the algae and identification and data analysis. For identification, the samples of macroalgae that have been sampled are placed in a millimeter box and documented using a camera coupling output. And for the data analysis, the species was collected were analyzed by using Shannon-Wiener diversity index, and the diversity index was computed and analyzed using the water stability table. Next. Okay, the sampling method, using systematic sampling method for collecting sample, and for calculating index of species diversity using Shannon-Wiener formula, and the diversity index were calculated by the formula that shown in the slide. And next. Okay, we move to the result and discussion. In this research, there is 22 macroalgae species were found consisting of 9 species classified into Chlorophyta, 3 species classified into Phyophyta, and 10 species classified into Rhodophyta. And the first species is Chaetomorpha crassa, that has a hair-shaped green alga that has characteristic rapid growing, hardy, and possesses a bright red to greenish-white appearance. And the second species is Enteromorpha intestinalis, has a tubular, roughly tactile organism that is 10 to 30 centimeter in length. And the third species is Bergensenia forbesi, that has a bubbles that sometimes also pear-shaped, has a bright green to yellowish green color, and the skin is thin, so the entire bubble is translucent. And the next species is Ulpalactuca, that has epious variants, shades of green ranging from light green to dark green, depending on its habitat, whether submerged or along the shoreline. And the next species is Caudoporosis javanica. The Caudoporosis genus is separated into the Caudopora 1 because it's branching of side branches and the multinuclear character of the individuals. So the cladoporsis resembles light green, compact cushion, and is common in the wave-breaking region. The next species is Prunetella nitida. The characteristic of Prunetella nitida is that it grows attached by small ridges like hopeless to rock and dead coral blocks, which are covered with thin sand. Okay, next slide. Next species is called tomentosum that has a thallus that has dichotomous structure, much branch with thin branches, each with a circular cross section. And next species is Acruma denticulatum that has a body shape resembling a leaf with long and flat leaf blades. Next species is Acanthora muscoides that grow between 6 to 16 centimeters tall with reddish to purple color. It attached to surfaces using a flat piece like hourglass and has abrasives. And next species is Calidia careosa that has a reddish brown color resulting from variation in light intensity and the presence of certain chemical. And next species is Gracilaria salicornia. It has a body composed of fine filaments resembling branches, and the size of it ranges from 10 centimeters to 30 centimeters. And the next species is Calidium purpurescens that have a distinctive reddish purple color, and they typically grow in rocky intertidal cones or shallow subtidal areas with clear waters. And next slide. Okay, next species is Gelidium corneum that has a cartilaginous dark red suet with flattened branches with spoon-shaped bracelet.
I'm sorry, you have 2 minutes left.
Oh, okay. Okay. Maybe I will make it short. For the next species, it's Hanidia crispa and Laurencia burkinati, Laurencia papilosa, Palmaria palmata, and Padina minor. And next species, next slide, please. And one is species found as Sargassum turbinaris. Yes, Sargassum crassifolium, Cladophora columina, and Catomorpha antenina. Next slide. Okay, next. Based on calculation with the index of species diversity using Channel Weather formula, the results show the index of diversity of macroalgae in cubits is 2.47. It means that the level of species diversity is moderate, and based on the measurement parameters of environment, the result of measuring pH was 7.97, and for the water temperature is 26.8 Celsius degree, and air temperature is 22 Celsius degree, and salinity is 34 ppm. Okay, next slide is okay. The last part is conclusion. The index of diversity of macroalgae in cubits was indicate as a moderate, and there is a 22 macroalgae species were found in the zone of Kuku Beach, Gunung Kidul, Yogyakarta, and last is physical chemical parameters show that air temperature, salinity levels, and pH levels significantly affected the growth of marine macroalgae species. Next slide. Okay, this is the references that we use in this research. Okay, thank you for your attention. I will bring back to the moderator.
Okay, thank you, Lina, for the great presentation. Okay, the last one in this session is Nimade Udayana.
Is that okay that
Yeah.
I share my own presentation?
Yeah. Time is yours for Miss Made.
Oh, okay. Thank you for the opportunity. Good morning, ladies and gentlemen. I'm very pleased to see you all. Om Swastiastu, Assalamualaikum warahmatullahi wabarakatuh, Shalom, salam sejahtera for us and everyone. Allow me to introduce myself. My name is Imade Diwanita Sokawijaya, or you can call me Dita. I'm a student from Marine Science major, Faculty of Marine Science and Fisheries of Udayana University. Again, thank you so much for the opportunity. And today I'd like to present my research about spatial and temporal distribution of stranded dugong or dugong-dugong events in Indonesian waters. Next, please. Okay, the first part is the introduction. So the reason why I took this topic to the research, and as we know that dugong is a member of the Sirenia order and has an important role in marine life, especially in the seagrass meadow ecosystem. But since 2015, IUCN defines that dugong are on vulnerable status and also on last 20 years in Indonesia, it was found so many cases about stranded dugong, and now and until now, about dugong population members and the exact habitat in Indonesia are still unknown. Next, please. With that background, the purpose of the research is to determine the distribution of stranded dugongs in Indonesia coastal areas using spatial analysis and knowing the seasonal characteristic of the distribution pattern of dugong stranding in Indonesian coastal areas. Next, please. Okay, the second part is in October 2022 until January 2023. With the study areas is located throughout Indonesian waters. Next, please. For the collected data is sourced from online database called Whale Stranding Indonesia and from articles in journal that collected online, and also that data then validated with cases recorded by Kementerian Kelautan dan Perikanan Indonesia or KKP. In the data collected for 20 years from 2003 until 2022 by record the location and time of the cases. Next, please. For knowing the temporal distribution, it can use the seasonal analysis which done by correlating by 4. The first wave MEM seasons, and the third one is June, July, August, or the GGA season, and the last one is September, October, November, or SON seasons. Next, please. For the spatial distribution, can use the hotspot analysis, which can identify the concentration of the phenomenon spatially and statistically significant. This analysis use GTS or GI star methods for group the category of an hotspot and a cold spot using GI bin value. For the table can be used on the slide. It can be seen on the slide. Sorry. Next, please. The third part is the result. This is the map of the distribution of stranded dugong in Indonesia for 20 years with total 2007 case, with stranding incident mostly happen in the areas of Sulawesi to Papua with total cases is 66 cases. Next, please. Okay, for the seasonal characteristic of dugong stranding distribution, it found that on the DGF seasons, with the total of stranded cases is 42. For the MEM season, with 33 cases. Next, please. For the GGA season and SON seasons, with the same total, which is 26 cases. And next, please. For the hotspot distribution, we can see that on the GGF season, hotspot is located in Riau Archipelago, Nusa Tenggara Barat, and South Sulawesi, with level of confidence hotspot is 95%. And from the MEM season, The hotspot is located in North Maluku with level of confidence is 99%, and Riau Archipelago with level of confidence is 95%. Next, please. For the GGA season, the hotspot located in Central Sulawesi and Maluku with level of research, we can conclude that the distribution of stranded dugong often occurs in the areas of Riau Archipelago, Sulawesi Island, and Maluku Archipelago. And total of incident is in the west season or the DGF season, which the season with the highest incident with total cases is 42 cases. Maybe that's all about my research. I'm sorry if there are any mistake about my word in the presentation. Again, thank you for all of your attention. I return the time back to the moderator. Thank you so much.
Thank you for the presentation, Ms. Made. And now we continue to the discussion session. Yeah, because we have the limited time, I just open for 2 questions. Please raise your hand. Ya.
Bu, ada pertanyaan di bawah.
Ya, apa-apa.
Ya.
Di mana?
Di bagian mana?
Di mana? Mention your name, institution, and mention to whom your question is for. Okay. Hello, I'm Natia from Udayana University, and for this session, I want to ask for the number 4, Laila or Aisa. Aisa, yeah. For the question is for Aisa. Okay. For your research, has there been any research on this topic before? And if so, what makes this research different from previous research? Thank you. Okay, different research before yeah. What will make different? Yeah. Thank you, Atia. And then the second question comes from the text, the chat box from Mr. Sabto. Mr. Sabto to Aisha and Asna or Sista. Why not measure chemical parameters due to macroalgae need nutrients for growth? Thanks. Okay. To the presenter, you may respond the question, please. For the first, for Miss Aisa, please respond the Atia question, please.
Okay, thank you for the question. I'll try to answer the difference about our research and the others. It is because we conducted the research in this year 2022. Yes.
Okay, on that. And
Okay, sorry.
yeah.
Okay. It is. Okay. Okay, I will continue on my answer. The difference between our research with others is because we conducted this research in this year 2023, and it can be to like yes it sorry. It is my voice.
I can't hear your voice clearly.
Okay, sorry. Okay. For make it short, it is because we never Usually we always make a research about diversity from year to years, and this is for 2023. And what it makes it species that we found. There is a new species before from the previous research, and maybe for my answer, it is enough for me.
Ms. Aiza, thank you for your answer. Okay, for the second one, the question comes from Mr. Hato. For Laila, maybe, or Asma. Yeah, for Aiza or Asma, please answer the question. Hello, Miss Aiza, Asna, or Aiza, maybe. Aiza, Asna, or Aiza. Yeah.
I'm sorry, for me, I'm not researching about microalgae, maybe Arvista.
Please respond the question.
Sorry, okay. Let me answer the question. First, because our physical-chemical parameter that we already use in this research is already sufficient for the research. And for several parameter like dissolved oxygen, we cannot measure it because we have limited equipments, so we cannot measure all of the parameters. It's all for me.
Oh, because the limited money.
Yeah.
Okay, thank you for the response, Ms. Laila and Hasna. Thank you. Arvista. Thank you. One question. Okay. Okay, I will read the question. To Miss Miss Diwianita, why is it necessary to do a hotspot analysis if you already get the spatial distribution result at the first? Okay, Miss Made, please respond the question.
Okay, thank you for the question. Why is necessary to do a hotspot analysis if we already get the spatial distribution or by the compiling the collected data? So the hotspot analysis can use to see more detail about where's the area that many cases about stranded dugong, and it can in later we can focus on that area to minutes about the incident about stranding dugong in for that cases. Maybe is that can answer my question?
Miss Made for the question. For the answer, I'm sorry. Okay, I'm sorry because we have limited time. I will close the session and we will continue to the next session. Please be applause for us for this session. Now we are going to continue to next session. In this session, there are 3 presenters. Yeah, the first presenter is Sekar Larasati, and then continue to Pierre Sandita Altavio, and then the last one is Muhammad Agung Triuda Agustiana. Okay.
Apa Mas ngajar?
Yeah.
Plotting apa
For the
KKN?
first presenter.
Kan baru UAS. Oh, mantap. Wis selangkah lebih maju. Wakeh soalnya Prodin.
I'm sorry,
Iya.
please mute your.
Ngeluh ya.
I'm sorry for the okay. I will continue. Now, the first presenter is Sekar Larasati. And is your Miss Sekar. Miss Sekar.
Thank you, Miss Moderator. Good day, everyone. Assalamualaikum
Itu
warahmatullahi
orang Banyuwangi itu, orang Amerika.
I'm sorry, Mr. Hato.
Assalamualaikum warahmatullahi wabarakatuh. Selamat siang, Om Swastiastu. My name is Sekar Larasati, and I'm from the Research Center for Limnology and Water Resources, BRIN. So I and my colleagues would like to present about our study, and the title is Molecular Markers Development and Validation for Anguilla Bicolor Detection Using eDNA Assay. Next. I don't see my slide.
Now, with the operator, wait a minute. Slide presentation.
Soal air ya. Kalimat.
Can you see the slide presentation?
Oh,
Diajukan ke manusia.
no, it's me. We should share screen.
Can you
Dari
see
Unas saya
the slide presentation, Mrs. Sekar?
Should I share my presentation slide?
Yeah.
Okay. Just a moment. Yes, I will do that. Just a moment. Could you see my presentation slide? Okay, I will start again. So my presentation and they are economically important. They are exported to several countries. However, the population, their population stage is near threat. So they're also important from the conservation point of view. So there should be a proper management for protecting and improving their sustainability in the wild, which usually include the routine monitoring. However, routine monitoring requires extensive field work, great effort, and the capture-based method that we use is sometimes not standard. So nowadays, people are using eDNA or environmental DNA assay, just like Professor Kim explained this morning, which can be used to detect and monitor the organisms, including one of the approaches of eDNA assay in this method, there are several steps needs to be done. So the first thing to do is we need to design primers and probe, which are specific to our target, and then we need to test their specificity in silico or bioinformatically, and in vitro in the lab, or and also in the field or in situ, or in the aquarium or in the pond trial. So once the primers and probe are validated for their specificity, they can be used in eDNA applications in the field. So our objective here is to develop and validate the specific primers and probe for detecting A bicolor eDNA. So the first thing to do is we need to design the specific primers and the probe. So we align sequences of Anguilla bicolor and other ungulate species, and then we need to choose specific regions for the primers and probe, and then we in the NCBI to see the list of species which can be amplified by these primers, and we want to have the list only A bicolor. And then after that, after we choose, after we have these primer candidates, and then we will go to the lab and non-target DNA using conventional PCR and visualize in the agarose gel. So based on the in silico test, we had, based on this, based on this in silico test, we have like these 27 primers candidates, and it showed that they are not subspecies specific. So these primers, we aimed actually in the beginning to target Anguilla bicolor, but then the in silico test showed that these primers could also amplify Anguilla bicolor pacifica. So, but the probe we designed was subspecies specific. It was only for detecting A bicolor. So from these 27 primers, we choose 3 candidates to be tested in vitro. And then we come up with this one candidate, we code this the candidate with this number, which passed the in vitro test and showed that these primers can amplify specifically to A bicolor samples. At this time, we only had A bicolor samples, A marmorata, and we had also tissue samples or DNA samples from other fishes. But here we show that the primers, they can amplify only to A bicolor samples. As you can see, these visible bands here only from the bicolor samples, this one as well, and no visible bands in the Anguilla marmorata or other fishes. And then we got samples from our colleagues in Sulawesi, thanks to Bapak Fadli Tantu, who sent us A bicolor pacifica tissue samples and also A celebesensis. So then we could test our primers against these species. And this in vitro test confirmed that the primer candidate was primers candidates were not subspecies specific. And after that, we went to the Aquaria trial with the questions, can the primers and probes specifically amplify A bicolor eDNA from water samples? So we took water samples from water tank filled with A bicolor. We also tried to put one A bicolor in the aquaria. In the aquaria, also one A marmorata. And then from this aquarium, we let them stay for 7 days. And then after that, we filter the water and extract the eDNA. And we did the qPCR assay. And we detect the presence and absence of A bicolor eDNA signals by looking at the CT value. So this is the results from the Aquaria trial. The first Aquaria trial. So you can see this purple curve is from DNA from the tissue samples of A bicolor. and it has the lowest CT value compared to the other samples. This one is from the water tank. These two are from the water tank, and this one is from the aquarium. And it's reasonable, of course, because DNA from the tissue is usually the concentration is higher than in the water. But here the results show that the primers, they can amplify eDNA from eDNA of AB color from the water samples, both from the tank and from the aquarium. And none of eDNA signals from the negative control, which is from the A marmorata, was detected. So the primers and probe can specifically amplify AB color eDNA. And then we also test with AB color Pacifica tissue samples in the qPCR assay, just to prove if the design probe can increase the specificity to detect only AB color bicolor. But apparently here we could see an amplification curve with this CT value from the AB color Pacifica sample. So we could also confirm that these design primers and probe, they are not subspecies specific. And we went back to the aquaria trial with the questions: can we relate the eDNA concentrations with fish density? So we had aquarium without any fish and aquarium with 1 and 6 AB color bicolor. So we did as the previous aquaria trial, but this time we measured the eDNA concentration. So here, here is the result. We could see that eDNA concentrations from the aquarium with 6 AB color bicolor is higher than the aquarium with 1 AB color. So here we could say that eDNA concentration is related with fish density. And then from the aquaria trial, we want to get closer to the nature. So we tried our primers in the from water samples from the AB color bicolor ponds. So there are several ponds near Ciberem River in Cilacap. So it's a AB color bicolor farm. And then from these locations, we took water samples from 3 ponds which have AB color bicolor inside. And then this is just to show our filtering device in the field. And this is to store the filter. And then after that, we brought them to the lab and measure and detect the presence of eDNA signals of AB color bicolor from the ponds. So here you could see that this is from the positive control, which is always high concentrations DNA. And these 3 curves, they are from 3 different ponds. So it's actually good. These primers, they can also amplify AB color bicolor eDNA from the ponds. So the eDNA of AB color can be detected from pond water samples. Then from the pond, we move to the water samples from the field. With the questions, can we detect AB color eDNA from river water samples? And then we took samples from the rivers, the 2 rivers flowing into Segara Anakan, here Ciberem River and Citandui, also from the lagoon and from the river mouth. However, we have not succeeded to detect eDNA signals from these locations. So we are still optimizing the qPCR assay to get the eDNA signal. We thought about the PCR inhibitor and we want to repeat the assay, but however, we are running out of the reagent, so we are waiting the reagent to come. To conclude, the primers and probe for AB color eDNA detection have been developed and validated in silico, in vitro, and from the pond water samples. However, the specificity test with other species, for example, with A nebulosa, because according to the literature, A nebulosa live in the same habitat as Anguilla bicolor. So the specificity test with this A nebulosa still needs to be confirmed. So if the audience here or your colleagues are working with A nebulosa tissue samples, maybe I could ask for a help to get a little bit of the tissue samples, so I can try my primers. And from this study, it also showed that eDNA concentration seemed to be correlated with fish density, and the validations from the field sample is still ongoing. And hopefully we will get positive results from our field sample in the future. So that was all my presentations, and thank you for listening.
Thank you, Ms. for the great presentation. Yeah. Okay. Now we continue to second presenter. Is Firsandita Altair.
Hey, can you hear my voice clearly?
Yeah.
Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh. May peace be upon us all. A pleasant afternoon to all of the audience in this meeting room. Good afternoon. Okay. Have conducted regarding the diversity of I do research on Kuku Beach. Kuku Beach, located in Gunung Kidul, especially reefs, are quite abundant. It ranges from sand to rocky reef and supports a diverse range of plant animal communities. The marine habitat within the seabed between the highest high water mark and the lowest low water mark is defined as the intertidal zone. Animals that live permanently in the intertidal zone have a variety of anatomical, physiological, and behavioral adaptation to help them survive in this difficult environment. Fish are found to have greater flexibility in dealing with these stresses, as they have greater motility than most other intertidal animals, and they use one of two basic strategies. Fish have an important role in the intertidal zone. This important role plays a role in the food chain and trophic levels in an aquatic ecosystem. The unique character of the intertidal zone of Kuku Beach is essential in studying its diversity, as there is no data regarding the fish in intertidal zone of Kuku Beach. Therefore, this paper aimed to determine the diversity of fish species in the intertidal zone of Kuku Beach. So we will move on to the methods part. This research was conducted in Kuku Beach, which is located in Kamadang Village, Tanjung Sari District, Gunung Kidul Regency, Yogyakarta. Sampling was carried out 2:30 until 4:30 a.m. on October 29, 2022. The tools used during sampling were nets, teaser, searing, ruler, millimeter block, pH meter, thermometer, refractometer, bucket, ziplock tray, and jar. Meanwhile, the materials used were alcohol 70%, water, and seawater. This research was distinguished into 4 phases, namely capturing, mounting, preservation, and identification. Capturing or sampling consists of random revolving sampling. Capturing fish during low tide, in which tide pools were visible. Fish were taken in the intertidal zone, specifically in the tide pools. This technique is classified as a random revolving sampling technique, as certain species of fish were not targeted. During capturing, physical-chemical parameters were also measured, such as water temperature, air temperature, and water pH, with the aid of the physical-chemical equipment. After capturing, the captured fish or specimen were anesthetized in 70% alcohol and seawater in a ratio of 1/3 in the tray of the specimen. Afterward, they were placed onto a millimeter box as a reference and comparison of their size between other specimens, followed by a recording taking pictures of the specimen. Once analyzed, the specimen were preserved in jar filled with alcohol and water in similar ratios, and the specimen were brought back to Yogyakarta City, where they were identified as performed with the aid of the FAO research platform and the record length, observing the shape of the fins and mouth and observing the body shape as a reference. From the following procedure that had been performed, it was determined that 4 distinct families were identified, namely Blanidae, Gobiidae, Muranidae, and Pomacentridae, along with 8 distinct species found in the intertidal zone of Cuckoo Beach. The first specimen is Coryphoblenus calidata, also known as the Montague's Blany. The latter line with vertical pairs of ports are found as well. There were white spots on the body that were evenly distributed, along with 4 until 5 brown spots in the middle of the body. The second specimen is Blania gibiform. The Blania gibiforms are characterized by a white yellowish body visible from the dorsal area of the body and black spot on the interspinous membranes of the first and second dorsal fins. Next is Asterogobius caninus, was identified well, characterized by a long and compact body, a flat head anterior to vertical gill openings, and scaly operculum. Bathygobius coletus was found in a rock pool, where the water reaches only during the highest high tide. Inhabits clear coastal reflux in intertidal to about 5 meters by 6 until 2 dorsal spines, 9 dorsal sulpharies, 1 anal spine, and 8 anal sulpharies. Next. Achitana bullosa, also known as the snowflake eel, is characterized by large mouth and numerous teeth, often with fangs, measuring between small medium within 3 meters of maximum size. Next, Buduefdif vagansis is a tropical marine fish with a yellow band around the base of the dorsal, 4 on the trunk and 1 on the caudal peduncle. Next is Stegasus obretus, has a moderately deep body and terminal, slightly oblique mouth. Juveniles have a yellow body color, narrow dark scale margins, and a blue spot on the head. The last one is Pomacentrus brachialis, inhabits acropora coral habitats. Pomacentrus brachialis is dark gray to blackish dorsal fins with black spots covering the base of the pectoral fins. Next. Aside from species identification, physical-chemical parameters were measured, including the water pH, water temperature, and air temperature. It was observed that Cuckoo Beach has a water pH of 7.73, the water temperature of 24 degrees Celsius, and air Celsius may have promoted their growth, as well as marine fish tend to have a higher growth rate in temperatures range degrees Celsius. So Cuckoo Beach having a water temperature of 24 degrees Celsius, it may have been the optimal condition for its growth. The highest of family is an algae eating herbivorous fish. So many of them are found on the coast of that's because they have lots of hole and crevices. The number of on the species of modernity they found. Next. According to the finding of the study, there were 8 species of fish in 4 families found at Untuk Beach. Pomacentridae was the most common family discovered, while Murinidae was the least common, as evidenced by inability to identify one specimen. There are still many possibilities and potential for different types of fish in the interior zone of Untuk Beach. Further research is required for better data collection as well as a variety of other purposes such as conservation and taxonomy. This is the end of the presentation. Thank you for your time and attention. If you have any relevant question, please ask. I return the session to the moderator. Thank you.
For the great presentation. Then we continue to the last presenter in the session is Muhammad Agung Triyuda Agustiana.
Assalamualaikum warahmatullahi wabarakatuh.
We will show you the video, the presentation of the Muhammad Agung Triyuda Agustiana. Okay. For the program, please.
Assalamualaikum warahmatullahi wabarakatuh. Budaya Fisheries for all the past. The macro-algae represent the subject of this study are best there to read in the in the food chain macro-algae and what is I mean what the shell and the sources what the first for the time. Because we trade and some again like balance so this come with using which is effectively does not occur and we balance yes, the top balance So this study aims to look at the condition of macro-algae because of the classification after to study how the exploitation is too developed on both coast for body infection. This region will firstly mention what carried out in application. We call it the dry system and also in the wet system because algae are liable which community structure can be referred to different systems. For important macroalgae ideas and also we don't or as herbivore can help develop the growth of macroalgae through two mechanisms, namely the activated growth of macroalgae. We conducted a study regarding the association between these two biota on the surface of West Java, which showed that the relationship between macroalgae and macrozobentos is in a positive direction, indicating that there is a beneficial relationship and also within the habitat variable. The correlation that we obtain is at the level of a medium positive relationship, where this relationship is arranged based on the relationship between the species cover with the species number of macrozobentos and macrozobentos density. The highest correlation occurred at the current application during the dry season, which during this season, so a high number of macroalgae species which might occur due to the warm water condition as described by Udayana, where the high abundance and distribution of macroalgae is more likely occur in the warm season. However, have a high level of accumulation of species compared to the salang-elang. This is thought to be due to the more stable characteristics of the substrate. Macroalgae that live by attaching to the substrate are very dependent on the type of substrate available in the location. The following table describes the substrate analysis carried out at two locations, where the Lantai beach is a rocky beach. Judging from the type of substrate, it is included in the category of gravel and coarse sand, while the rocky beach at Lantai is included in the category of coarse sand and other substrate mixture. These differences will greatly impact the presence and cover of macroalgae. Throughout the season, Lantai was included in the location with the highest number of species possible with the characteristic of the beach substrate discovered earlier. This allowed more macroalgae to attach and avoid being hit by waves. The species Phalaenopsis vaccinella, Urvala punta, and Gracilaria canabiculata are the species that are consistently present in this study, where the species belonging to the class of Chloridiophyceae and Butrophyceae tend to live on rocky substrate and also in mixture of stone and sand. As previously explained, horizontal transect laying was carried out to see the gradual differences in macroalgae vegetation from the shoreline to the beach edge. Lantai has a higher number of species on the shoreline and is the opposite of silahkan. The condition on silahkan beach can be explained by the recent of home, where macroalgae in the intertidal zone are spread from the top of the intertidal to the bottom of the intertidal, and it is possible that the number of species found in the bottom of the intertidal is still quite high. Observation of macroalgae cover, which is the variable that determines the correlation carried with the previous macrozobentos, shown that in terms of cover, the wet system has higher opportunities that were under, including drought which occur in the dry season can reduce biomass, so that it linearly reduces macroalgae cover as well due to the season that will affect the biomass of macroalgae. In terms of location, the cover on silahkan beach is higher than that of Lantai due to the high cover of the Floridiophyceae class, which morphologically has several species that have wide size. Judging from the cover condition from the shoreline to the beach edge, the silahkan location so then the condition of other aquatic biota observed was macrozobentos, where the number of macrozobentos species in dry season was higher than the wet season, which was made possibly by the high number of macroalgae species in the dry season. The presence of macrozobentos is strongly influenced by the distribution of macroalgae, where the high number of macroalgae species allow for a greater diversity of morphology macroalgae species, thereby phylum mollusca is often found in this study, which show the good adaptability of this phylum to environmental dynamics. The species often found in this region is Diadema or sea urchin, which makes sunken soon, which is quite abundant in this region as a food source. The density of macrozobentos on the seashore is higher than on the beach edge. which is caused not only by the high number of macroalgae species in the seashore, but also because of the more protected condition from macrozoobenthos from the crossing water. Sayang Helang has a higher macrozoobenthos density than Karapia, which is supported by the high macroalgae cover in this location. In the end, this study concluded that the abundance of macroalgae species in a location is a factor of the number of macrozoobenthos species. A higher density of macrozoobenthos is expected in the dry season, which is in line with the abundance level of macroalgae species during the same season, other than the higher density of macro is only attributed to the higher density of the macroalgae cover. The value of correlation coefficient determine that macroalgae and macrozoobenthos in this study fall into medium category with a positive value for the association value. Thank you.
Yeah, thank you for the video presentation from Mr. Muhammad Agung Priyuda Agustiana. Okay, now we continue to the discussion session. There are one question for Mrs. Lucia and three questions for Miss Lucia. For the first question is from Mr. Sabto. Mr. Sabto from silahkan.
Yang pertama
dari why you use specific primer? Okay. For Miss Silahkan, please respond the question. Hello, Miss Silahkan.
Yes, I'm still here.
Yeah.
So why am I using the specific primer? Oh, but Bapak Sabto, it is nice to meet you. It's nice to meet other researcher who works also in eDNA. So maybe we can share experience, and maybe in the future we can collaborate. Why am I using the specific primer? Because I want just targeting A bicolor. Well, in the end, it is A bicolor. That's why we need to design the specific primer, because it should amplify only A bicolor, and they should not amplify other ungulate species. And with these specific primers, or the target species assay of target species approach of eDNA assay, we can use PCR. Probably it will take several days to get the data. While using the qPCR, you could get only 2 hours. You can find out whether this A bicolor is there or not. And also, we can also measure the eDNA concentration. So you can one day in the future estimate the abundance or biomass of the target species. So that was all the answer. Thank you.
Thank you, Mrs. Saka, for the great answer. And then we continue to Miss Lucia to respond the question from Miss Dianova. Yeah, the question is there any fish within the same species but slightly different in body morphology? Yeah, is there any fish within the same species but slightly different in body morphology? That is the first question. And then the second question is, what are the relationships between niche and Antarctica fish? And the last question is, why sampling was carried out in the early morning? And the fourth question is, do you make data analysis of this biodiversity? Okay. Please respond the question, Miss Lucia.
Okay, thank you. The first question is from Mr. Sabto. Thanks for Mr. Sabto's again. Sorry, however, for now, we only do which species are found on the surface biodiversity in Coco Beach. Thank you. And the second question is from Miss Deanova. In our family, we found one species with a marked difference in coloration, namely Daniela gibboniformis, wherefore which may indicate a genetic variation within the same species. Additionally, several pieces of literature report with white-colored Venice with black markings. However, the species found in Coco Beach were found to be dominantly gray or black, which may further indicate a form of genetic variation or perhaps a camouflage habit the Venice process when under stress. And the third question is from Miss Violeta. Tide pools or niche are breeding areas for many fish species. Within tide pools, there are rocks which can be used as shelters for many intertidal and subtidal invertebrates. So niche or tide pools are an important part of coastal and marine fish population, as this habitat is part of the home range for several species. In a tide pool composed of sand, rubble, barrow, and rock covered with algae. And the last question is from Ms. Revita. Sampling was carried out in the early morning because where it was still dark due to the low tide, revealing a series of tide pools due to their ability to avoid predators and maintain good image quality in scopic vision as a result of an evolutionary syndrome of several morphological and physiological traits. Reef or tide pool fish are found to be relatively active in the dark or night. Thank you. This is my response. I return the decision to the moderator. Thank you.
Thank you, Ms. Lucia, for the great answer. Okay, I'm sorry, the discussion time is over. We finally come to the end of our session. On behalf of the committee, I would like to thank the presenter for the insight and input from participants during the session. Please give a big applause for. Thank you for the great work, and I will see you in the next event. Thank you. Good afternoon.
My name is Muhammad Agung Triuda. As
Oh, yang Kak Ella? Jadi apa itu? Iya. Ya, Pak Alam, kita ke Suci. Oh, itu yang kita tanya itu langsung. Suci dia bilang sama saya, Rintan, kayaknya itu nih caranya Pak Dedi supaya dia kasih pindah saya dari WD2. Saya bilang, kenapa? Dia bilang, Pak Alam, Suci, jangan mau cerita apa-apa, karena takutnya nanti Pak Dedi sudah takutkan kok bocor sama ibu-ibu dan wanita di luar yang kegiatan-kegiatannya kita tidak boleh ke orang luar. Tanya, oh iya Pak, siap. Dia bilang,
aduh, itu bocor suaranya Ibu Yustika. Suaranya kemana-mana, maaf.
Iya, terima kasih Bu, mohon maaf.
Nggak apa-apa, mungkin kepencet kali ya Bu.
Iya, kepencet tadi Bu, mohon maaf.
Good afternoon to all presenters and seminar participants. It is an honor for us to have you all here. First of all, let me introduce myself. My name is Ayu Putu Wiweka Krishna Dewi from Udayana University. I will be the moderator in this session. Ladies and gentlemen, now we will try the video presentation. After the video has played, we continue to the discussion session. In the first session, there are 4 video presentation. Namely, the first presenter, the second presenter is from Bahia. The third presenter is from Arista, and the last presenter and the last video is from Zairon. Okay. For the first video presentation is from Umi Prodija. Yeah. Please play the video. Video.
Assalamualaikum warahmatullahi wabarakatuh. I want to presentation my paper with title Some Indicator for Assessing Stock of Retail Scat Characteristic Rates in Palabuhan Ratu, West Java, Indonesia. By Mufadriah Tirtadanu Prihatiningsih, Mufauzi Karsono Wagio, and Yoga Hani Restiangsi. Introduction. Red tail scab, or the Captaris curatus, is one type of small pelagic fish that has high economic value is dominant. This study aims to determine several indicators for assessing stock red tail scab in Palabuhan Ratu waters, including language relationship, sex ratio, spawning adverse maturity, land adverse capture, growth, and mortality. It is hoped that the result of this study can be used as a reference for consideration in sustainable fish resources management and as an effort to monitor retail scat fish stocks. Material and methods. The study was conducted from February to December 2019. 2,813 samples was collected at fish landing site at Palabuhan Ratu. Result and discussion. The results showed that the distribution of the total length ranged from 14.7 until 45 centimeters, with average of 25.7 centimeter. The growth pattern was positive allometric, which means that the length increase of the rate of red tail scab is faster than the weight gain. The sex ratio of red tail scab is not balanced. Based on information of the gonad maturity stage and gonadal somatic index GSI, it can be said that the peak of the red tail scab spawning season in Palabuhan Ratu waters occurs in May until November, and the peak occurs in July. The land adverse capture of the red tail scab is 26 from gillnet, 23 from handline, 26.7 from bottom longline. Land adverse maturity is 25.8. Size at maturity is influenced by multiple factors, including genetic difference, environmental variable, fishing pressure, and food availability. The result of population dynamic analysis for red tail scab showed that the value of asymptotic length and growth rate was 25.23 centimeter and 0.61 per year respectively. The fishing mortality 0.91 per year, natural mortality 0.81 per year, while the exploitation rate 0.52. Conclusion. The peak spawning season of red tail scab occurred in July. The use of gillnet and bottom longline fishing gear is more selective than handline. The fishermen can catch red tail scab fish a minimum size at 26 centimeters. Utilization of red-tailed catfish in Pelabuhan Ratu is likely higher than optimum or is fully exploited. The management must be based on sufficient scientific data by considering the country of fishing activity of the fishermen. The management must be based on sufficient scientific data by considering the country of fishing activities of fishermen, as well as the looking for alternative activities other than fishing that can be sought as a compensation for closing session, or reducing effort. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, thank you for the first video presentation. The second video is from Bastian.
Oke, kata itu kita udah mau pakai alias mistika.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Thanks very much for your attention. Today I want to present you our poster about study of growth and condition index of freshwater pokea clams Bathysa violacea variant celebensis in La Solo River Estuary, Southeast Sulawesi by us, Bahtiar, and me, Yustika Intan Permatahati, Ermayanti Ishak, and Latifa Fikri. Pokea clams are freshwater clams that inhabit river mouths and can be found in almost all major rivers in Southeast Sulawesi, including the Laya River, Langkumbe River, Pohara River, and La Solo River. These freshwater clams possess important economic value and are cultivated by fishermen, particularly toward larger-sized clams with large biomass, can result in a decrease in the growth of the clam and the condition index of the clams. However, the study on the growth and condition this study aimed to assess the growth of pokea clam species in the La Solo River, Southeast Sulawesi, through its length-weight relationship and condition index. Method: the present research was carried out for a year in January until December 2014. In the sample clams were then counted, and their length and weight were measured using a caliper and a digital scale with an accuracy of 0.5 mm and 0.01 gram, respectively. The clam sample were brought to the laboratory and separated by sex based on the color of the gonad, whereby male clams have milky white color and female clams have a yellow to brown color. And this is the result of the pokea clam of length-weight relationship in La Solo River, Southeast Sulawesi from January until December. Clams were 2.52 and 2.70, which indicate the dams had negative allometric growth. The low B value of pokea clams in the La Solo River can be assumed to be the result of the hack exploitation of the clams, which favors clams of larger size and mid quantity. The hack exploitation would cause a shift in the size of clams available in nature to smaller sizes, thus producing low B values. The condition index values of male and female, respectively, 3.98 and pedal feeding mechanism, which is allow pokea clams to have a high productivity. The second, the La Solo River receives supplies of nutrients from fertile soil upland, which is highly favorable for the growth of the clams. And moreover, the condition index can also be influenced by the thickness of the clam cells, whereby clams with thicker cells tend to have smaller index values. And the conclusion that the Pokea clams in the Lasal River had a negative allometry with the condition index being in the medium to fat category. Moreover, the Pokea clams undergo gonadal development in February and partial spawning until October. That's all our presentation.
Thank you for the video presentation. And the next video presentation is from Mira Astawidia.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. My name is Mira Aristawidia. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. My name is Mira Aristawidia, a student from the Department of Aquatic Resource Management of IPB University. Thank you for giving me the opportunity to Indonesia. My background research was due to various activities around the waters of Situ Gunung Putri that might input waste such as heavy metals into the waters. Heavy metals pollution in the waters can certainly threaten the health of ecosystem and human health. Therefore, this study aims to determine the distribution and accumulation of heavy metals in the waters of Situ Gunung Putri from the water, sediment, and heavy metal content in the fish. The research was carried out from August to December 2022, including water quality measurement in Situ Gunung Putri and samples analysis at the Research Center for and water located near the inlet from the ex-agricultural and Station 4 located near the outlet. The methodology consists of water quality measurements as well as analysis of heavy metals, and then the analysis of bioaccumulation factors was done to compare the content of heavy metal in fish with the heavy metals in the water. The concentration of heavy metals measured in water tends to be low at several sampling stations, but the highest concentration of lead and zinc at Station 4 or the outlet have exceeded the quality standards of PP number 22 of 2021. Meanwhile, all the heavy metals in sediment at each sampling station have exceeded the threshold effect level, and the highest concentration of each heavy metals was found at Station 1, which is located near the inlet from the industries. The low concentration of heavy metals in the water while high in the sediments is suspect that there is an absorption of heavy metals by the water hyacinth at a fairly fast rate, because the water hyacinth is found in these waters in very abundant quantities, especially at the Station 1, where the cover by water hyacinth is denser than at other stations. The fish obtained from sampling consists of Oreochromis mozambicus, Pangasius species, and Hypostomus species. The Oreochromis mozambicus samples had variations in body length, so the samples were divided into 4 groups to see the difference in heavy metal accumulation based on difference in body length. The heavy metals in fish meat show that lead and zinc in all fishes have exceeded the quality standards. Copper concentration is still safe, while high concentration for chrome found in the smaller fish, and the concentration of cadmium has exceeded the Indonesian national standards. In some heavy metals, it can be seen that the graph decreased in Oreochromis mozambicus as the length of the fish body increased, which is indicate the elimination of heavy metals in the body as the fish gets older. The research showed that the fish are still dangerous for consumption, with the detection of several heavy metals in the fish meat that exceed the quality standards, so the accumulation of heavy metals will be very dangerous for the food chain and for the human health. and then bioaccumulation factor showed a value of more than 1,000 for copper and zinc in each sample of fish meat, even though the concentration of copper in fish meat was still below the quality standard. A bioaccumulation factor value of more than 1,000 can be interpreted as a potential accumulation for the copper and zinc in the fish meat. For the conclusion that can be drawn from this study is that the highest concentration of heavy metals in the sediments at Station 1 shows that effluent from industry can increase the heavy metal concentration in the water, and then the content of several heavy metals in fish meat, especially lead and zinc, has exceeded the threshold allowed for fish consumption. Thank you for the attention. Good afternoon.
Thank you for the video presentation. And for the last video presentation in this session is from Udayana.
Good morning, everyone. I am Udayana from Faculty of Fisheries and Marine Sciences, IPB University, Indonesia. First of all, I would like to say thanks to committee who gave us a chance to present the result of the study. On behalf of our team, let me present our small research with the title Biological Reproduction Variable of Spiny Lobster Panolerus homarus linnaeus 1758 in west coast of Lampung province. The spiny lobster Panolerus homarus is one of economically important marine species in Indonesia and intensively caught by the fisher in almost its distribution area, including in west coast of Lampung. Research on the biology and population dynamic of spiny lobster have been carried out in some Indonesian water. However, little information on the biology of this species as a fundamental for management in the west coast of Lampung province, whether the spiny lobster population in the area as one stock unit in the Indonesian Fisheries Management Area 752 or not. This study purpose was to analyze the biological reproduction variable of spiny lobster, for example, sex ratio, language relationship, and relative condition factor, maturity, and reproductive potential. The spiny lobster sample as a dominant catch was 21 to March 2022 using the random sampling method. The data collecting included sex, total length, carapace length, total weight, gonadal maturity level, fecundity, and condition of gonad in female carrying egg or buried female. Gonad maturity set were classified based on Silva and Dalla Cruz Landim 2006 and also Kabiddu et al. 2008. Results shown that spiny lobster carapace length was in the range of 41 to 94 millimeter. Sex ratio was relatively balanced. Negative allometric growth pattern for both male and female. Condition factor as well as gonadosomatic index tend to be increased from October to December for both male and female and then decrease. Gonad with fully mature stage or stage 3 found every month for both sexes, and the peak of spawning season during observation period occur in November to December. Size at onset of spawning at 63.10 millimeters carapace length, while fecundity is as reproductive potential.
Thank you for all video presentation. Now we continue to the discussion session. Please raise your hand, or you can write in chat box. Yeah, any question for the presenter?
apapun.
Okay, before
Hello.
yeah. Yes, please.
Yeah.
Mention your
Roma.
Oh yeah.
Hello.
Yeah, Roma, please, and your question and mention to whom your question is for. I'm sorry, Miss Tia Roma, unmute your device, please.
My poster presentation have to Mr. Agung.
Okay. Your video presentation will be played after the session.
Okay, I have a question. Can I ask?
Yes. You
Okay.
can answer the question. You can ask the question for the presenter first.
Okay, thank you. Hi, my name is Henry TLOK.
Oh
I have
my
question for Miss Mira Aristawidia.
Yeah.
Oh, I'm sorry if I say wrong your name. Very interesting research. You found that PB, CD, and ZN over the threshold in fish meat, right? So I just want to know, how do you do your research? Which part of fish that you sample, and how did you do the research? I mean, like, measure the chemical in the fish body. That's all.
Okay, thank you for the question. For Mrs. Mira Aristawidia, please respond the question.
All right. Thank you for the question. Is my voice clear?
Yeah.
Yes. Yes. That's fine.
Firstly, I apologize. I have a problem with my camera, so my face isn't showed. Okay. Thank you for Miss Henderit. How can I call you?
Please call me Henny.
Henny.
Yes.
Okay, thank you, Miss Henny. Okay, your question is about how I did my research and which part of the fish I use for my sample. For the heavy metals analysis, I use the etching method. So I use the meat part, only the meat part of the fish. I fillet the fish and then I burn the fish to ash and then use the acid destruction with nitrate acid, and then I measure it with AAS.
Okay, so it's all part of the body or just certain part of the body?
Just the meat where we can eat it.
Oh, okay. So every meat in fish body you take it, and
the
then
fish, the meat was diaduk until
okay.
homogen, and then I analyze the sample.
Oh, great. Okay, thank you.
Yeah.
Thank you for the response. And then maybe there are any question? Okay, I have one question for Mrs. Mira. Yeah. Because the result is very interesting. Yeah. I want to know what activities motivate of the content of heavy metal in the study site. What activities motivate?
I'm sorry. Do you mean the industry activities or?
No. What activities activities most effect human activities around the site most affect the content of heavy metal?
The most activity that impact the heavy metals in the waters might be from the industries, which is there is a lot of industries around the waters, and also the settlements that might input the heavy metals to the waters, such as the industries maybe paper industries and manufacture that use metals in the process, and then automotive like that.
Okay.
Is my answer clear?
Yeah. There's human activities in the around the site.
Yeah.
Yeah. Oh. Okay, thank you, Mrs. Mira, for the response. I think that's all for the first session. Please give big applause for the first session. Okay, we are going to continue to the second session. Yeah. In the second session, there are four presenters. Yeah. Namely, the first presenter is Mrs. Sugiarti, and then the second presenter is Mrs. Yahroma Husnina Nasution, and then the third presenter is Mrs. Hendrik L.O.E., and the last presenter is Mr. Iman Supriyatna. Yeah, stay with the first session. We will share the video, and then after the video has done, we continue with the two discussion session. Okay, for the first, we will play the video presentation from Mrs. Sugiarti.
Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera untuk kita semua. Thank you for the time. I would like to present our research with the title of Preliminary Study of Water Quality Analysis in Upper Citarum Watershed, East Java, Indonesia. We are Sugiarti, myself, Yoyo Sudarso, Aiman Ibrahim, Muhammad Yamin, Muhammad Jamroni, and Laila Sari. We are all from BRIN. The background of our research is Citarum River is one of the longest and largest rivers in West Java province. The length of the river is approximately 30,000 kilometers with a watershed area of 600 kilometers. The population in the Citarum River catchment area reached 15,000 people. Anthropogenic activities have a major contribution pollutant load to the river. The Citarum River pollutant sources come from agriculture, household, industry, and other productive service business. If the condition of a river is damaged, if it's not treated quickly, it can reduce water quality, ecology, function, and river ecosystem services. The Citarum watershed still requires monitoring of water quality, since it is suspected that Citarum River is still polluted due to the pressure from human activities. The aim of this study is preliminary research of water quality monitoring in the upstream Citarum River to be the basis for watershed management. It also useful to know the water quality for fisheries. Research was conducted in 8 sampling stations in the upstream Citarum River, namely Gunung Wayang, Cikitu Village, Babakan Village, Wangi Sagara, Puncak Cahe, Koyot, Andir, and Nanjung. We can see in the sampling site figure we do sampling from the north to the south. From station 1 to station 8. We can see the figure from station 1, station 2, station 3, station 4, station 5, 6, 7, and 8. This is an analytical result. Post-sport analytical result. Other result analysis. And PCA result. There are 3 groups. Group 1 is station Puncak Cai and Gunung Wayang. Group 2 is Cikitu Village, Babakan Village, and Wangi Sagara. That group is Koyot, Andir, and Nanjung. So the conclusion is most of the analytical results still meet the quality standard. The result of the PCA analysis showed that there were 3 groups. Water quality monitoring of Citarum River must be carried out continuously to evaluate its water condition.
Yeah, thank you. That's all the video presentation from Mrs. Sudiati. Now we continue to the next video presenter is from Mrs. Tia Roman Kusumi Nasution.
Introduction University will be presenting our paper entitled Several Ecobiological Parameters Billy Mystacologus padangensis as an endemic fish in Lake Singkarak, West Sumatra. This lake, Singkarak and Billy fish. The lake is tectonic lake, Solok and Tanah wide. The lake 11,200 hectares and depth 295 meters and in depth 158.2 meters from our research on 2022-20. Billy is an endemic fish is an important economic value in Lake Singkarak as the lake is a recreation park, hydroelectric plant, and ecotourism as a source of water for domestic uses. Lake Singkarak has a unique and endemic fish and species as Billy is one of the attraction of this lake. Billy fish has important economic value, so the prices is quite high. Billy fishing effort continues to increase every year. The reduced production of catches and the smaller size of the Billy fish catch so indicated that the Billy fish population in Lake Singkarak is starting to be threatened. The aim of research was to examine some of the ecobiological parameters of Billy fish. The method of the samples was collected from 8 stations Sumpur, Center Hydropower Intake, Paninggahan, Moropingai, Sumani, Aripan, and Ombilin, and Batu Taba. Sampling location were selected based on the characteristic of the habitat. The fish capture with experimental gill net, 3 mesh size. Water quality analyzing uses the Horiba water quality checker and determination of grain size of the sediment carried out by dry shipping method. And the result of this research, the highest frequency of bellyfish length was found in the 75 to 81 millimeter class interval at Sumani Station, with an average fish length of 78.1 millimeter, while the highest fish weight in the class range of 15 to 23 milligram was found in Paninggahan, an average fish weight of 19.1 milligram. And positive allometric growth patterns were found in Batu Taba, Intake PLTA, Paninggahan, and Sumpur. And while negative allometric growth was found at Ombilin, Sanyang Baka, Sumani, and Sumpur. And the highest gonad maturity stage 4 in male fish found in Sanyang Baka, and the female bellyfish was found in Sanyang Baka and Ombilin 100%. The gonad maturity index value in males is 2.2 to 10.1%, while the gonad maturity index in female is 6.4 to 15.6%. The water quality in Lake Singkarak temperature, pH, dissolved oxygen, millimeter class interval. The highest fish weight in the class rank 15 duration in Sanibaka. The characteristic of
for the devil cichlid Amphilopus labiatus in Lake Sentani, Indonesia. Freshwater ecosystem in Papua, the Indonesian half New Guinea, have begun experiencing a broader range of introduction of exotic species over the last decade, including Lake Sentani. One of the dominant and abundant exotic species in the lake is Amphilopus labiatus. Research was undertaken to document the length-weight relationship and the condition factor of this fish within Lake Sentani. Fish were collected using floating gill nets between June to August 2019 at 6 locations from southern part to western part of the lake. Red devil cichlids Amphilopus labiatus were collected and counted. Furthermore, 20 individuals were measured for their total body length, body depth in millimeters, and body length per month, which described by the equation Kn equal to W square AL square of W divided by AL square B. Key results of our research were the fish growth pattern of Amphilopus labiatus is positively allometric, with B values ranging from 3.19 to 3.20, and the coefficient of determination ranging from 0.84 to 0.87. Although mean length of Amphilopus labiatus males was less than the females, condition factor values were not significantly different between sexes, ranging from 0.64 to 2.03 in males and 0.67 to 1.19 in females. However, in month-by-month condition factor data, there is a marked reduction in condition factor in August, indicating a spawning event in late July or early August. Conclusion of our research was Amphilopus labiatus is the most abundant and dominant introduced species in Lake Sentani at the time of the study. We confirmed Amphilopus labiatus grow well and occupied most habitats in Lake Sentani. That's all I want to share. Thank you very much for your kind attention. Thank you very much. Research and Community Service Directorate, Research and Technology, Higher Education Ministry of Indonesia, who funded me to do the research in 2019. Thank you very much, my students from Biology Department, Mathematics and Natural Sciences Faculty of Cendrawasih University, who helped me to do the research. And thank you very much, villager from the 6th location of Lake Sentani, who helped me to do fish collection. Thank you.
Thank you. That's all the video presentation from Mrs. Hendrik L. Okay, next for the last video presentation is from Mr. Iman Supriyatna.
Okay, I'm sorry. Before, first I apologize. I sent a video in the PPT, but I don't know because my voice recording is not clear, so I choose music. Sorry.
Yes. Sorry, Mr. Iman. Pardon me.
Yes. Please, the music, the video can.
Yeah, I will play your video.
Yes. Thank you for committee and all participants. My name is Iman Supriyatna from Fisheries and Marine Science Doctoral Program Berwijaya University. I will present about article review about gastropoda as behavior of microplastic. As we know, issue and problem related to the impact of microplastic have generated significant public concern from primary and secondary sources. Maybe over 5 trillion plastic debris and 200,000 million tons of plastic waste floating around the ocean, including on sea ice, and the economic cost about $3,000 per ton. The meaning of microplastic, I think, is too many. The video is too many face.
Okay.
I'm sorry. Yes.
Yeah. You can explain your poster. I'm sorry, Mr. Iman.
Okay.
Do you want to continue explain your poster?
Yes.
Yeah, you can continue with this slide. Yeah.
Okay. The study study have discussed about microplastic in aquaculture area, and also the organisms are at risk of exposure to microplastic. So anticipatory monitoring and prevention efforts are needed to detect the level of microplastic pollution in the water of aquaculture area. In here, bioindicator is meaning that species are increasingly being used in monitoring microplastic pollution, and gastropod species are good candidates for this role. And biocontrol are living organisms that are used to evaluate the health of natural ecosystem in the environment. Also, the study state that gastropoda as microplastic indicator agent in aquaculture area. Also, bivalve shellfish and gastropod as the indicator and biomonitoring agent for aquatic environment pollution. Sorry, this is a review article presentation. For more information, maybe next you can see our link about our article at the following link that I can share. Sorry, maybe that's my presentation. Thank you.
Okay, thank you for our presenter. The all of video presenter has done play, and then now we continue to discussion session. I will open just one session because we have limited time. Okay, raise your hand and mention to whom the question is for. Any question? Okay, maybe I have one question for Mrs. Tiaroman. Yeah. It's very interesting research. Yeah. My question is, what are the possible long-term plans to maintain the breeding fish population in that location? Do you hear my voice, Mrs. Tiaroman?
Could you like to remain your question, please?
Yeah. My question is, what are the possible long-term plans to maintain the breeding fish in your location, in your reservoir?
Not clear your voice. I'm sorry, not clear your voice. Not clear. your voice?
Yeah. What are the possible the possible long-term planning planning long-term plans to keep the population in this population in the same location.
Oh my God, your voice is not clear for me. Maybe you can write in chat.
Yeah. Maybe we have a question for Mrs. Oki.
Yes, please.
Yeah. My question is there negative impact from the existence of living fish for the ecosystem in around the site location?
Oh, okay. Okay. The negative impact of Amphilopus labiatus is not studied yet, but day by day, I observe that Amphilopus labiatus is really bad for native. Some native species are carnivore fish. They eat Amphilopus labiatus, and they're like it goes to the throat and then stuck there, and it's kill the fish. So many native fish, especially Octodron heterodon, one of the native Gedgen in Lake Sentani, have really serious problem with this fish. This is the first one. And the second one, Amphilopus labiatus is growing very fast in the lake. So the first time I found this fish in 2008, 2009, around there. So it grows very well, very fast. The abundance is really high now in Lake Sentani, and it's really problem for native who occupied along the beach, like rainbow fish, another native species like Gyuris margaritaceus, the fish where they occupied along the beach. So it's really bad impact for the ecosystem as well as the native species.
Okay, thank you, Mrs. Kia.
No problem.
Well, the disease
Your voice is not really
Yes, not clear. I'm sorry.
Can you repeat?
I'm sorry. Not your voice not clear, intermittent.
is introduced to the Lake Sentani. How to there about existence in Lake Sentani.
Who did the introduction to the lake? Okay. Oh, there is no data who did it. Everybody wants to be the bad guy. So I asked to the fishery department, they said that they don't do that. So there is no data who introduced the fish to the lake. So it just like how I say, show in 2007, 2008, 2009, and nobody knows who introduced this fish to the lake. As just for information, that we have more than 50% of fish community in Lake Sentani are the introduced fish. So it's really big problem. This fish is one of the most abundant and most dominant fish in the lake right now.
Because the problem maybe comes from restocking, yeah. Sometimes there in the restocking event, we don't care about the specific species, yeah, or introduce the lake.
Yeah, it's really hard to understand your statement because your voice is your statement is not clear. Anyway, it's big problem in Lake Sentani, and we still struggling how to control the fish in the lake. Thank you.
Yeah. We need control to the for the restocking fish maybe, yeah. Okay.
Maybe.
Yeah.
Yeah, maybe. Yeah. I believe it's one of the action that we can do in Lake Sentani. Thank you.
Okay. I will to answer your question, Miss. What are the possible long-term plans to maintain the billyfish population in that location? I think to maintain the billyfish population to continue, and in this Lake Singkarak, we can to conservation and to manage the fish, because the fish is endemic fish only in the Lake Singkarak. So we can to maintain the billyfish to continue and the population with conservation. I think this maybe.
Thank you, Ms. for your question and for your answer. Because we have limited time, and this session is over. Yeah, the time is over. So I will close the session. Yeah. Honorable presenters and participants, we finally come to the end of our session. On behalf of the committee, I would like to thank the presenter for the insight and input from all participants during the session. Please give a big applause for all of us. Okay.
Thank you.
Yeah, thank you for the great work, and I will see you in the next event. Thank you. Yeah, there is information from the committee. Please back to the main hall for continue the next agenda. Thank you.
You're welcome. Thank you. See you soon.
Yeah, thank you.
We can leave to main room.
So we have to leave to main room, or we can leave.
Or
we
can hear.
Yeah, all the presenter can back to main hall.
Oh, bye.
Yeah.
Okay, thank you.
Yeah, back to main hall. Main hall.